Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Charles Bernard. He's a PhD student at Sorbonne University. And we're going to talk about quorum sensing, but quorum sensing perhaps from viruses. So this will be really cool.
Exactly.
Charles, thanks for coming.
Yeah, thanks you for having me. It's a real pleasure.
Yeah, tell me about your research, please.
I'm a PhD student, and my PhD project is about microbial communication. And what we mean by microbial communication is a biological process called quorum sensing. Basically, when you look at the dictionary and you look at the quorum, the definition is the minimal number of people that is required in a room for a decision to be taken. So if you translate it in the world of microbiology, it's the minimal number of bacteria or viruses that needs to be there for a collective behavior to be efficient and therefore to be triggered. And basically, what I'm trying to do during my PhD is to answer the following questions. Who communicates, with whom, how, and to which ends, basically.
When you say who communicates with whom, so from what I understand, like in our guts at least, you have fungi, yeast, protists, bacteria, viruses, phages. I mean, so what, if I dropped you in there, and I miniaturized you, what would it be like?
Exactly, because in fact, in the microbial world, we know for a fact that many different species use quorum sensing. You are right, fungi, bacteria, and recently in 2017, a team in Israel, Erez and co-worker, discovered for the first time that viruses also can communicate, and it was a very major breakthrough in the world of microbiology. Yeah.
Well, tell me about that study. What did they look at? What did they find?
Yeah, so here the virus that we are talking about is a bacteriophage, which are the viruses of bacteria. And in bacteriophages, there are some kind of bacteriophage that are called temperate bacteriophage. And basically, what we need to understand is that a virus cannot self-replicate. So in order to replicate and to spread in nature, to survive, it needs to infect a bacterium. But the thing is that when it infects a bacterium to replicate, it kills the bacterium. But as a free form, a virus cannot survive longer. It needs to be within a bacteria to be somehow protected. So if a virus keeps replicating at the expense of the host, and if the host disappears, then the virus cannot survive. So temperate bacteriophage have also another life cycle, which is the lysogenic cycle, in which the viruses inserts its genome into that of its host and confer upon its host immunity toward its free form. So this lysogenic cycle is a cycle that protects the phage host collective and then prevents the host population from extinction. So we have the virus, the lytic cycle, during which the virus replicates fastly at the expense of the host. And you have the lysogenic cycle, during which the virus protects the host, but replicates much slower as part of the bacterial genome. And there is a trade-off between fast replication at the expense of the host and also the need to protect the phage host collective. And basically, what they have discovered is that this decision to transit from the lytic cycle to the lysogenic cycle was controlled by communication. In fact, viruses communicate to estimate the number, and when the viruses are extremely numerous, then the host cells are likely to be few, and therefore the lysogenic cycle is triggered. So if you want, I can talk.
Okay. Yeah. So the viruses are, once they're inside their cells, they're able to count and see how many of them there are, and how are they doing this? Are they causing the cells to release like extracellular vesicles with this information, or how's it done?
Yeah, in fact, the way they are communicating, they use two genes, a gene that produces a signal and a receptor that recognize the signal. And basically, each individual virus of the population of viruses will produce this signal when it infects a bacterium. So basically, the more viruses there are, the higher the concentration of this signal. And therefore, the concentration of this signal produced by the viruses reflects the density of population of viruses. So in fact, at a threshold concentration of this signal is imported within bacteria and starts to be recognized by its receptors, which trigger the transition from the lytic cycle to the lysogenic cycle. So when the viruses are many, they stop killing their host and they start protecting their host with the lysogenic cycle. And it was
But again, how is it produced? What does the signal look like? Where does it come from? Where does it go?
Okay, so the signal is carried by a gene, which is translated into an mRNA and then into a protein. This signal is processed by the cellular machinery of the cell. And this peptide is then exported by the infected bacteria. So when a virus is inside an infected bacterium, the cellular machinery of the bacterium will produce the genes of the viruses that encode the signal, and then we'll export the signal into the medium.
Okay, and then obviously virions are not picking it up, but other cells are picking it up. So when you say exported to the medium, is it like a plasmid that another bacteria picks up?
In fact, the signal, which is a small peptide, is exported into the medium. And you're totally right. Other bacterium will import this signal when it is at high concentration. And then within infected bacteria, the signal will bind to the phage encoded receptors, which will trigger the transition from the lysis to the lysogenic cycle.
Oh, okay. And what about in animal cells or in human cells? Is it packaged in a vesicle? Like, how is it picked up?
I'm not an expert about quorum sensing in eukaryotes, but usually in bacteria, this signal is either a small peptide, which is exported by protein that triggers the export of the peptide towards the medium. And this signal molecule can either be a small peptide or a metabolite, a small small chemical molecule. And at high concentration, this signal is imported by a permease, which is a protein at the membrane of the cells.
Right. So when there's a lot of infected cells or a lot of virions floating around, I would guess the criteria is a lot of infected cells. Then it goes into more of a lysogenic phase, right?
Exactly. Yeah, because the higher viruses, the higher the concentration of this signal, and at a threshold concentration of this signal, it is robustly imported within all bacteria, and within infected ones will transduce the transition from the lytic cycle to the lysogenic cycle. So suddenly, within the population of viruses, they stop killing their host and will start protecting their host because the viruses are many and the host cells presumably few, and therefore the phage host collective is endangered. And the viruses somehow has to protect its own life by protecting its hosts.
Well, okay. So is it just about number or is it about other factors? Because viruses can be latent, inside of a bacteria for a long time. And then when host conditions change, then they turn lysogenic. So maybe there's other factors in addition to just the number of them and the concentration.
It depends also the type of viruses that we're talking about. Some, for instance, just trigger the transition from the lytic cycle to the lysogenic cycle in a stochastic way, which is randomly, basically. But in certain bacteriophages, this transition is controlled in a density-dependent manner, which is a more optimal way to control this transition. And basically, this communication just control the transition from the lytic cycle to the lysogenic cycle, but not the other way around.
Oh, not the other way. How do you know not the other way around?
We don't know yet, actually.
Okay, but it just hasn't been discovered, right?
Yeah, well, yeah, exactly. Perhaps it exists, but so far it's not discovered.
Well, what might be interesting is to look at signaling in a system like Vibrio, where it can acquire a certain piece of phage DNA or phage nucleotides and then become cholera. What kind of signaling is going on to have that happen or not have that not happen, or antimicrobial resistance too. Like, what kind of signaling goes on to stimulate take-up of phage DNA,
In Vibrio, there is a very interesting discovery that has been made in 2019. Is that, as you may know, Vibrio use quorum sensing to estimate their own density. So this bacterium use quorum sensing to know when there are many, and then to switch from a individual behavior to a collective behavior. And there are some phages that are able to recognize the quorum sensing molecules that these Vibrio bacteria use to communicate. So basically, when the bacteria are many, which is reflected by high concentration of the communication signal that they are producing, there are some bacteriophages that can integrate this information, and then we'll start the lytic cycle. So in fact, the phage in this case has only the receptors that allows to recognize the phage, the signal molecule produced by its host. So it's kind of eavesdropping, if you want, or espionage. And so basically, the viruses integrate the information that the host are many, and so therefore they can kill the host without killing the host population.
That's interesting. So, viruses must have some sense of self versus other, because the quorum sensing goes on for their particular, if you want to call it species or set of quasi-species. But what about in superinfection environments or environments? The viruses are doing quorum sensing, but just again for their own kind. But what about other viruses that may be in the area?
You mean communication between distinct viruses?
Yeah, different kinds of viruses. If if a certain virus is taking up shop, and then another virus comes in and seems to start taking over or proliferating, what's that communication look like? Is there an alert where the virus has to step up protection? What if it's in a lysogenic phase, but it needs to literally step up protection in order to protect the host?
In fact, communication between viruses is a very new field of research, so we don't know much yet about it. And so far, we don't know if different kind of viruses are able to sense each other's by eavesdropping communication molecules. But perhaps it exists actually. So far, the quorum sensing systems that have been discovered in bacteriophage are extremely specific, which means that each bacteriophage speaks its own secret language. But perhaps there are other bacteriophages that are not discovered yet that speak perhaps a language which is less secretive, more universal, and then can be captured by other bacteriophages. But we don't know yet this.
Well, bacteria communicate with other bacteria, right? To trade resources and metabolites, and they communicate with our own cells, right?
Yeah, this is for sure that bacteria can communicate with themselves, with their own kind, but can also communicate with other species and can somehow contextualize their own density relative to the density of the microbial community. In bacteria, this is known, but in viruses, it is not known yet. But perhaps it exists as well.
Interesting. Are you involved in studying any other communication, or is it just purely viral, or what's the focus of your study?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Lately, I've been working mainly on new tools to discover novel communication systems within bacteriophages. And because communication systems, they have some features, some genomic features that I try to understand. Once I understand them, I can develop models in order to detect this kind of signatures and try to find them as well in bacteriophages. And I've been developing such kinds of methods recently, and I've discovered novel communication systems within bacteriophages. And I also discovered such kind of novel communication systems within a bacteriophage that already had another communication system. So basically, I discovered somehow the first phage with two different quorum sensing systems. So the first phage that is able to speak and understand two different languages. So that was a new discovery that I made recently.
What do you mean? So it speaks. It's able to speak to its own kind, but what other species can it speak to?
The virus?
Yes.
So far, we don't know if it's able to speak with other species. When I say two languages, in fact, it produces two communication signals that accumulate differentially in the medium, which means that if you want, the threshold concentration of the first signal would be reached, for instance, before the threshold concentration of the second signal. So the two communication systems will somehow help the phage to know different regimes of density. So very low, middle density, and very high density. And then between these different regimes have different actions that are well suited for its survival.
So, okay.
A bit like a developmental program, if you want.
Can scientists make any of these signaling molecules and dope a system, where like there's bacteria that's infected and they're able to produce these signaling molecules and just dump them into the system and see what the effect is? If you flood the system with these, will all of a sudden the phage change activity and go lysogenic? I mean, it could be used maybe as a therapy. If you've got some kind of infection and you find the signaling molecule and you put it into the person, let's say, in high amounts, and it fools the phage, fools the virus into thinking that tons of cells are infected, and then maybe it goes lysogenic or it goes latent. That may be a therapy.
Yeah. Basically, we are more and more studying quorum sensing as a mean of therapy. Also because bacteria, for instance, this is known that bacteria become virulent at high density, and they use quorum sensing to estimate their own density and to switch from an individual behavior to a collective virulence. Today, we are studying some therapies, for instance, to try to break down the signal that the bacteria produce to estimate their own density and to become a pathogen. So this is called Antichrome sensing. And so basically, and also what is important is that I discovered a quorum sensing system in phages, which controls not phage processes, like the transition from the lytic cycle to the lysogenic cycle, but that controls the expression of the genes of its host, presumably. So basically, at low density, for instance, the genes of its host would be repressed, some genes of its host, and at high density of phages, this inhibition would be alleviating. So somehow, these communication systems could help a phage to manipulate its host in a density-dependent manner.
Very interesting. So if there's a large concentration of this signal, and the phage goes goes latent or it goes lysogenic, you see the production of virions like go way down. And I mean, like what do you see? You said it's protective of the bacteria, but how so? Like what behaviors do you see change?
Exactly. It's not me, but these are people that studied that. Basically, you see, for instance, the growth of the bacterium that starts to increase after the lytic to lysogenic transition has occurred. During the lysogenic cycle, bacteria are being killed, so the growth curve decreases. But when the switch from lysis to lysogeny is triggered by viral quorum sensing, then the bacteria starts to grow again, and you can really see these curves. And basically also the number of virion drops as well, because they cannot survive longer without. All the phages within bacterium become lysogenic, and the free phages cannot survive longer without being inside the host. So you could as well see the decrease of population of virions.
Oh, okay. So you see a cluster of phenomenon is what you're saying.
Yeah, exactly.
Okay. where are you going to take this? The fact that there appears to be quorum sensing, and this is governing the behavior of viruses. Like, again, what's your take on it? Do you think this would be made into a therapy, or what do you think is the highest and best use of this?
Yeah, for me, it's really interesting the fact that viruses can manipulate their host in a density dependent manner. And what is really interesting is the phenotypes that these viruses can influence, the phenotype of the host that the viruses can influence in density dependent manner. Because we've seen that, in fact, we predict that viruses can communicate to estimate their own density and manipulate the sporulation or the biofilm pathway of their host. And it is really interesting because, in fact, it is as spores that bacteria mostly become infectious. For instance, you have many bacteria in your gut, and these are anaerobic bacteria that are really sensitive to oxygens. And in order to transmit to other people, they have somehow to become spores to resist air exposure and are able to spread and to infect other people. So if you have phages that somehow prevent this bacteria to sporulate in density-dependent manner, that's really interesting from a therapeutical point of view. You could think, for instance, of introducing phages that could somehow inhibit the sporulation of their hosts and therefore counteract nosocomial infection or stuff like that.
Well, if a virus goes into a spore formation, then it's going to be inactive, right? Or I'm sorry, the bacteria does.
Yeah, the viruses becomes inactive, but the spore, its biology is inactive, but it's often as a spores that a bacterium colonize other niches, and therefore it's the infectious forms of many bacterium, the spores. For instance, Clostridium difficile, which is a pathogen, it is as a spore that it becomes infectious and it is responsible of nosocomial infection. So if you have some phages that prevents these bacteria to become spores, that can be really interesting from a medical point of view.
So you think the phages are governing the behavior of the bacteria, whether to form a spore or not?
My feeling, I don't know if it's governing, but it's clearly influencing, modulating. And that's really interesting because the current paradigm is that a bacterium becomes a spore in an adverse situation. But let me ask the following question, like, who gets to decide when it's time to sporulate? Who gets to decide whether the situations are adverse or not? We used to think that these are totally the bacteria, the genetic systems of bacteria that decide this. But in fact, we realized that, maybe these are the phages that communicate to estimate the density and manipulate the host biology accordingly. So somehow you can think Phage like a puppet master or something like that.
Well, we don't know who calls the shots. I've seen examples of bacteria taking parts of a phage genetic material and using it, like expressing their own spike proteins to poke other bacteria. But then you see phage using bacteria for their own ends too. So it's like, who calls the shots and when?
Definitely, you have exchange of genetic material between bacteriophages and bacterium, because during the lysogenic cycle, the phage inserts its genome into that of its host, and so the genes of the phages are provided to the bacteria during the lysogenic cycle. But you can also have the converse case, which is the copy of genetic material from the bacterium to the phages. And lately we've done an analysis, which is the phylogenetic tree of a communication systems that regulates the sporulation. And in fact, we can see that these systems, which is shared between bacteria and viruses, is frequently exchanged between viruses and bacteria. So in fact, this ability to control the sporulation in a density-dependent manner is currently exchanged between these two microbiological entities and shape the biology of each other. So many communication systems are exchanged between bacteria and viruses, and this is in shaping the biology of the different microbiological entities.
I don't know, what's ahead for your work? What do you think that you're going to be able to figure out possibly in the near term?
In the future?
Yeah, specifically for your research, what questions do you feel like you're closing in on? What do you want to get clarity on in the next year or so?
Definitely, I want to characterize more communication systems of bacteriophages that are known to control the host in a density-dependent manner, because this is really brand new. This was never discovered before, and trying to understand better the dynamics of these systems. And I'm also working on another project which consists in identifying all the genes of communication within genomes of bacteria, viruses, etc., and then compare these genes of communication across genomes to see which genes are similar across genomes, and then to infer which microorganisms are susceptible to communicate likewise. Because if they have similar communication genes, somehow we can infer that they are able to produce a similar communication signal, and therefore to be able to communicate together. I will use this information to construct a network to infer which microorganism is susceptible to communicate with whom, which bacterium is susceptible to communicate with which other bacterium, which other viruses, and then to comprehend somehow the role of communication in ecology, etc. So trying to build the first map of what's going on in the microbial world in terms of communication, a communication network, if you like.
Okay. Well, what's the best way for people to find out more about your work, Charles? Where can they go?
You can go on the website of my laboratory, which is the Team R, AIRE, and it stands for Adaptation, Integration, Reticulation, and Evolution. And here you will find the publication and what my lab is doing. Okay,
one last thing in that acronym, reticulation. What does that mean?
Reticulation is basically what gives rise to network structure, so interaction. And in fact, interaction is at every level of biology, interaction between molecules, between cells, and between species. And basically, in my lab, we use network sciences, which is the science of interactions to understand biology and evolution.
Okay. Well, very good. Charles, thank you for coming on the podcast. I appreciate it.
Thank you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.